When you think about microbes, you probably picture them out in nature, right?
Yeah.
Soil, oceans, maybe forests.
Mm-hmm.
But what if I told you some of the most powerful ones are actually, well, engineers living inside the places we build,
That's right. They're doing some pretty incredible things, like helping us get metals out of rocks or cleaning up pollution.
but also influencing, the air in our homes, the water in our pipes? It's kind of wild.
It really is. And that's what we're diving into today, the microbiology of the built environment. It's this whole hidden world that affects our infrastructure, our health,
Mm.
sometimes good, sometimes not so good.
Yeah, we've looked at a whole stack of sources on this, trying to piece together how these tiny organisms play such a big role in our human-made world.
And our mission here is really to unpack that for you. We want to cut through the noise and give you the essential insights, the most interesting bits about these invisible inhabitants and their impact, help you get up to speed quickly.
Okay, let's get into it. Maybe start with one of the most surprising uses. Microbes as tiny miners.
Yeah. Earth's greatest chemists. That's what some call them. We've actually figured out how to use them to extract metals from ores that are too low grade, too poor for traditional mining to be economical.
And this isn't some small-scale thing either, right? I read something like a quarter of the world's copper comes from this microbial leaching.
Exactly. It's significant. The process itself is, well, pretty clever. You basically pile up this low-grade ore in huge heaps, like massive dumps.
What?
Then you let an acidic solution, usually around pH 2, trickle down through it. As it flows, it dissolves the metals.
And you collect the metal-rich liquid at the bottom.
You got it. That liquid is then processed to recover the metal.
So what's actually doing the work inside that pile? It's not just the acid.
No, the microbes are the key drivers, specifically certain bacteria that love acidophiles, things like Acidithiobacillus ferroxidans and Leptospirillum ferroxidans.
Mhm.
And if parts of the pile get hotter, you might even find archaea, like sulfolobus, doing the job.
Right. But they're not just munching on the copper ore directly, are they?
No, it's more indirect, mostly driven by iron chemistry. Bacteria like Acidithiobacillus are amazing at oxidizing ferrous iron, that's Fe2 plus O, into ferric iron, F3 plus O.
Okay, so the bacteria make the F3 plus L. Then what?
Well, F3 plus is a really strong oxidizing agent. It chemically attacks the sulfide minerals in the ore, like copper sulfide, CuS.
Ah, I see.
And that reaction oxidizes the sulfide and releases the metal, like copper ions, Cu2 plus, into the acidic solution. The crucial part is that the bacteria keep regenerating that F3 plus from the Fe2 plus produced, so the whole cycle keeps going.
So the bacteria are like the engine constantly refueling the chemical reaction that releases the copper.
Precisely. They drive the whole thing.
And getting the copper out of that solution, you mentioned processing it.
Yeah, a common way is called cementation. You basically add scrap iron FeO to the liquid. The iron metal causes the dissolved copper ions, C2L, to precipitate out as solid copper metal, Cu. And bonus, this reaction produces more Fd plus stat, which often gets recycled back to the leach pile or ponds for the bacteria to oxidize back to F3 plus again. It's a neat loop.
And our sources said this isn't just for copper, uranium too.
Yeah, similar principle for uranium. The bacteria keep the iron oxidized to F3 plus stat, and the F3 plus then chemically oxidizes the uranium from an insoluble form, U4 plus stat, to a soluble form, U6 plus stat, which can then be leached out.
Okay. What about gold? That was mentioned, but it sounded a bit different, not these huge piles.
Right. Gold is often trapped inside other minerals,
Uh-huh.
like arsenopyrite. So you use similar acid-loving bacteria, Acidithiobacillus again, to break down that arsenopyrite and free the gold.
But not in a giant heap.
Usually not. It's often done in smaller, contained bioreactor tanks. You get really high recovery, like over 95%. And a big plus is that this process helps remove toxic stuff like arsenic and avoids using cyanide, which is common in traditional gold processing. So it can be environmentally better in that sense.
Okay, so microbes as miners, pretty amazing. But then there's the flip side, the sort of double-edged sword aspect, acid mine drainage.
Ugh, yes. AMD. This is where those exact same microbial processes, the oxidation of sulfide minerals, cause huge environmental problems if they're not managed.
And this happens when mining exposes minerals like pyrite, that's iron sulfide FeS2, right? Like in coal mines or metal mines.
Exactly. Deep underground, these minerals aren't really exposed to much oxygen or water. They're stable. But mining digs them up, brings them to the surface, or opens tunnels.
letting the air and water get to them.
Right. And when pyrite meets oxygen and water, a slow chemical reaction starts things off, producing some F2 plus bilophor ferrous iron.
And that's the food for those same acid-loving bacteria we just talked
You
about.
got it. Acidotheobacillus, leptospirillum. They jump on that F2 plus and oxidize it to F3 plus incredibly fast. Way faster than the chemical reaction alone.
Okay, so we get lots of F3 plus again, and that's the problem.
That's the main driver of the damage. The Fe3 plus reacts aggressively with more pyrite. This reaction turns out even more F2 plus for the bacteria, keeping the cycle going, but crucially, it also produces sulfuric acid, H2SO4.
Ah, so it's a runaway cycle. Exposed pyrite busts bacteria, make A3 plus really fast, F2 plus attacks more pyrite, generates more Fe2 plus fuel and sulfuric acid.
That's the destructive feedback loop. The sulfuric acid drives the pH down, sometimes incredibly low, like below pH 1.
Wow.
And the super acidic water then starts dissolving heavy metals locked in the surrounding rocks, iron, aluminum, cadmium, lead, you name it.
Creating this toxic soup that flows into rivers and lakes.
Killing fish and pretty much devastating aquatic ecosystems. It's serious stuff. You even find bizarre microbes like ferroplasma and archaea that can live at pH 0 in these conditions.
Which brings up that question again, why doesn't this happen naturally underground?
It really comes down to that lack of exposure, like our sources emphasize. Without mining disturbing things and letting oxygen and water flood in, the whole destructive cycle just doesn't get started on a large scale. It's human activity that unlocks the potential for AMD.
But then in another twist, we can also use microbes to try and fix these kinds of problems through bioremediation.
Yeah, bioremediation is essentially harnessing microbes to clean up pollution. Sometimes you stimulate the microbes already there, that's biostimulation, or sometimes you add specific microbes known to do the job, that's bioaugmentation.
Okay, so how do you clean up something like uranium contamination? You can't really destroy a metal.
You can't destroy it, but you can change its form to make it less mobile, less dangerous. The strategy for uranium is to convert the soluble form, U6 plus ISEG, which travels easily in groundwater, into an insoluble form, U4 plus S.
Locking it in place.
Exactly. U4 plus often precipitates out as a solid mineral called uraninite.
And there are bacteria that can do this reduction.
Oh, yes. Certain bacteria are really good at it, especially under anaerobic conditions, meaning no oxygen. We're talking metal reducers like Shimana and Geobacter, and even some sulfate reducers like Desulfovibrio. They basically breathe the U6 plus sen, using it as an electron acceptor instead of oxygen.
So you can encourage them by, feeding them.
That's the idea behind biostimulation. You inject an organic carbon source they can eat, like acetate, into the contaminated groundwater. This gives them the energy they need to respire using the uranium, reducing it, and immobilizing it. Field tests show it can work pretty well.
But there's a catch, right? With how long uranium lasts.
There is. The big challenge is long-term stability. If conditions change later, say oxygen gets back into the system, that immobilized U4 plus could potentially reoxidize back to soluble U6 plus, and given that U-238 has a half-life of 4.5 billion years, yeah, you need that immobilization to last.
Okay, shifting gears to organic pollutants. Oil spills, hydrocarbons, microbes can handle those too.
Absolutely. Many microbes naturally degrade hydrocarbons as a food source. Especially with oxygen around aerobic conditions, you get a whole community of bacteria, fungi, even algae that can break down oil components, ultimately oxidizing them to CO2.
That helps that process along.
Warmer temperatures are generally better, and they need nutrients like nitrogen and phosphorus, just like plants do. Some bacteria are specialists, like Alkanivirax. It thrives in oily environments and even produces natural detergents, surfactants, to help break up oil slicks and make them easier to access.
I remember hearing about natural degradation playing a big role after the Deepwater Horizon spill.
It did. The type of oil spilled there, with lots of lighter, more volatile components, was actually more amenable to natural breakdown. Chemical dispersants were also used, which break the oil into smaller droplets, increasing the surface area for microbes to attack, though the dispersants themselves raise other environmental questions.
But hydrocarbons aren't just spills. They can cause trouble inside things like oil storage tanks.
Definitely. In big tanks, especially where oil sits on top of water, you can get anoxic zones at the bottom. If there's sulfate present, either from the oil itself or the water, sulfate-reducing bacteria, SRBs, can grow. They munch on hydrocarbons and produce hydrogen sulfide, H2S.
That rotten egg smell.
That's the one. But H2S is also corrosive. This process, called souring, can eat away at the steel tanks, causing pitting and eventually leaks. Aerobic degradation isn't usually the main issue in sealed tanks because oxygen is limited. Okay, let's talk about the really tricky stuff. Synthetic chemicals, xenobiotics, things like chlorinated solvents, plastics, stuff nature never made.
Yeah, you'd think microbes would have no clue what to do with those.
It's surprising, but they often do find ways. Our sources point out that nature does produce some halogenated organic compounds, think forest fires, volcanic activity, even some microbes make them. So microbes have evolved enzymes over millions of years to deal with these natural organohalogens.
And sometimes those enzymes happen to work on our synthetic versions.
Sometimes, yes, or they can evolve further. But how well they work varies hugely. Some chemicals break down relatively quickly. Others, like DDT, persist for ages. And conditions matter: temperature, pH, oxygen, what other food sources are around.
Right, and sometimes the breakdown isn't complete or it needs another food source. What was that term? Cometabolism?
Cometabolism, exactly. That's when a microbe only degrades the pollutant if it's also munching on something else easier to eat. It sort of breaks down the pollutant by accident while focused on its main meal. But the danger sometimes is that cometabolism might only partially degrade the pollutant, and the intermediate chemicals produced can actually be more toxic than what you started with.
Oof. Okay. But there's a specific anaerobic process that seems really important for chlorinated stuff. Reductive dechlorination.
Yes, this is huge for dealing with nasty chlorinated solvents like PCE and TCE, common industrial contaminants, often carcinogenic, or things like PCBs. Under anaerobic conditions, some microbes can use these chlorinated compounds as electron acceptors in their respiration, like how we use oxygen.
So they breathe the pollutant.
Kind of, yeah. And in doing so, they pluck off chlorine atoms, releasing them as harmless chloride ions, basically, salt.
And that's better than trying to break them down with oxygen.
Often, yes. Aerobic degradation of things like TCE can be tricky. It's usually cometabolic, needs oxygen or methane injected, which can be costly and even create explosion risks. Reductive dechlorination is anaerobic, so you avoid those issues.
Is there a star microbe for this?
There is a bacterium called dihalococoids. It's kind of special. Many microbes can pop off one or two chlorines, but dihalococoid species are the only ones known to go all the way. They can take something highly chlorinated like PCE or TCE and strip off all the chlorines, eventually converting it to ethene, which is harmless.
Why is going all the way so important?
Because if the process stops halfway, you can end up accumulating intermediates like vinyl chloride, VC, which is actually more toxic and carcinogenic than TCE in some cases. So having dihalococoids there to finish the job is critical.
So how do you encourage dihalococoids to do its thing in a contaminated site?
It needs an electron donor, usually a hydrogen gas, H2. So a common strategy is to inject simple fermentable organic compounds, molasses, vegetable oil, stuff like that, into the ground. Other bacteria ferment these, producing the H2 that dihalococoids needs. You might also add nutrients or even add dihalococoids cultures directly if they aren't present. That's bioaugmentation. Okay, from toxic solvents to plastic. Another major xenobiotic headache.
Yeah, we make billions of tons that last forever, ends up as microplastics everywhere. Seems like a lost cause for microbes.
It largely seemed that way for a long time. But then came a really exciting breakthrough with pit plastic. That's the stuff most drink bottles are made of.
They found something that eats it.
They did. A bacterium called Ideonella sakaiensis. It was found living on peat fragments, and researchers discovered it produces two unique enzymes, pTase and MBTase. These enzymes work together to break down the peat polymer into smaller molecules that the bacterium can actually absorb and use for energy and growth.
Wow, that's incredible. Actual plastic-eating bacteria.
It offers some real hope, a potential biological route for dealing with plastic waste, though scaling it up is still a major challenge.
And there are biodegradable plastics, like PHAs, but they're just not as cheap as the regular stuff yet.
That's the hurdle. Economics still favors the persistent plastics for now in many applications.
Okay, let's shift from waste to something vital. Our water systems. Microbes are absolutely central there, aren't they?
Completely. Clean water depends on wastewater treatment, and that treatment relies heavily on microbial processes to remove pollutants, especially organic matter. We measure that organic load as BOD, biochemical oxygen demand.
How much oxygen the microbes need to break down the waste.
Exactly. And the goal of treatment is to slash that BOD way down before the water is discharged.
How does that work? It's usually a multi-step process.
Right. First is primary treatment. Basically physical removal, screens catch big stuff, then settling tanks let solids simmer out. That sludge often goes off to anaerobic digesters.
More microbes at work in the sludge digestion.
Oh yeah. Anaerobic digestion is a powerhouse of microbial activity. In the absence of oxygen, complex communities of bacteria and archaea work together to break down the organic solids. This drastically reduces the sludge volume, and a valuable byproduct is methane gas biogas, which can be captured and burned for energy.
Okay, so primary is physical, then secondary treatment tackles the dissolved organic stuff.
Correct. Secondary treatment is where aerobic microbes do the heavy lifting on BOD removal. The two main methods are activated sludge and trickling filters.
Activated sludge.
You basically bubble air through the wastewater in big tanks. This encourages microbes to grow in clumps called flocks, often held together by slime produced by bacteria like Zooglobia. These flocks munch away on the dissolved organics, getting BOD down by maybe 95%.
And trickling filters.
That's different.
Right.
You have beds of rocks or plastic media, and you let the wastewater trickle over them. Microbes form a slimy biofilm layer on the media surface, and as the water flows past, the biofilm bugs degrade the pollutants.
So secondary gets rid of most of the organic waste, but sometimes you need tertiary treatment for nutrients, like phosphorus and nitrogen.
Exactly. Excess nutrients cause big problems in rivers and lakes, algal blooms, dead zones. Phosphorus can be removed chemically, but that often creates toxic sludge.
So there's a microbial way.
Yes. Enhanced biological phosphorus removal, or EBPR. It uses special bacteria called PAOs, phosphorus accumulating organisms like a cumulobacter. You cycle them between anaerobic, no oxygen, and aerobic with oxygen zones. Anaerobically, they release phosphate but store carbon compounds. Then, aerobically, they use that stored carbon for energy and take up huge amounts of phosphate from the water, storing it inside their cells.
Clever, so they concentrate the phosphorus inside themselves.
Right, and you remove the phosphorus by removing that microbe-rich sludge. No toxic chemical sludge involved.
What about nitrogen? That usually starts as ammonia in wastewater.
Standard treatment is a two-step microbial process. First, nitrification. Aerobic bacteria convert ammonia to nitrate. Second, denitrification. Other bacteria, under no oxygen conditions, convert nitrate into harmless nitrogen gas, which just bubbles away.
But denitrification needs a carbon source, right?
It does. And if the wastewater doesn't have enough natural carbon relative to the nitrogen, you have to add some like methanol or acetate, which costs money.
Are there smarter ways to do nitrogen removal now?
Yes, several advanced processes. One is nitritation denitrification. The idea is to stop the first step, nitrification, halfway at nitrate instead of nitrate. You can do this by controlling conditions like temperature or sludge age to suppress the nitrate oxidizing bacteria.
Why stop at nitrate?
Because converting nitrate to nitrogen gas in the second step requires less oxygen overall and less added carbon compared to starting from nitrate, so it saves energy and cost.
I also saw mention of aerobic granular sludge. That sounds different.
It's a really neat innovation. Instead of loose flocks, the microbes are encouraged to form dense, compact granules that settle much faster.
What's the advantage of granules?
The key is their structure. Granules can develop an oxygen-rich outer layer, but an anoxic or anaerobic core. This means you can have aerobic processes like nitrification happening on the outside, while anaerobic processes like denitrification happen simultaneously inside the same granule.
Wow, so different processes in one particle.
Exactly. It allows for more compact treatment systems, potentially saving energy and space compared to systems needing separate tanks for aerobic and anoxic conditions.
Okay, one more thing from the wastewater side, that liquid left over from digesting sludge, the brine.
Right. The sludge dewatering creates a side stream, sometimes called brine or centrate, that's super concentrated in ammonia and phosphorus. Treating this efficiently is important.
And there's a special microbial process for that too.
Yes, the Animex process, often coupled with partial nitritation, PNA. First, you convert about half the ammonia to nitrite, partial nitritation. Then, unique Animex bacteria take over. These are anaerobic bacteria that can directly combine ammonia and nitrite to produce nitrogen gas.
So they short-circuit the traditional nitrification-denitrification pathway.
Pretty much. The benefits are huge. No need for an external carbon source, significantly less energy needed for aeration, less sludge produced, and lower greenhouse gas emissions. It's a game changer for treating high ammonia streams.
It feels like we're getting really good at removing the standard pollutants. But the sources mention these contaminants of emerging concern. What are those?
Yeah, that's the next frontier. We're talking about traces of pharmaceuticals, personal care products, pesticides, flame retardants, chemicals that get into wastewater constantly from household and industrial use. but that conventional treatment plants weren't designed to remove completely.
And even tiny amounts can cause problems.
They can. Things like synthetic estrogens from birth control pills, even at parts per trillion levels, have been shown to cause feminization in fish downstream. So, figuring out how microbes might help degrade or transform these emerging contaminants is a big area of current research.
Okay, so wastewater gets treated, discharged. Eventually, some of that water might become drinking water after more treatment.
Yeah.
What's the microbial angle there?
Drinking water purification is obviously critical. The main goals are removing pathogens, getting rid of unpleasant tastes or odors, clearing up turbidity, and removing certain metals or chemicals. It involves multiple steps.
Like letting particles settle out.
Right. Sedimentation first, then coagulation, adding chemicals like alum that make tiny particles clump together into flocks. Flocculation is gentle mixing to help those flocks grow bigger, then more sedimentation to let the flocks settle out.
And then filtering.
Yep. Filtration through sand, activated charcoal, maybe ion exchange resins removes even smaller particles, many microbes, and dissolved organic chemicals.
Followed by disinfection, killing off whatever microbes are left. Usually chlorine.
Chlorination is still the most common primary disinfectant. The hypochlorous acid, HOCl, it forms, is a strong oxidant that kills most bacteria and viruses. However, some pathogens are pretty resistant, especially the protozoan cysts like Cryptosporidium. Also, chlorine can react with organic matter in the water to form potentially harmful disinfection byproducts.
So alternatives exist.
UV radiation is increasingly popular. It uses ultraviolet light to damage microbial DNA, preventing them from reproducing. It doesn't leave a chemical residual, doesn't form the same byproducts, and it's very effective against things like Cryptosporidium, often used alongside or instead of chlorine.
And they leave some disinfectant in the water as it goes into the pipes, right? A residual.
Yes, that's secondary disinfection. Usually a small amount of chlorine, or more often chloramine, which is more stable but less reactive, is left in the water to help prevent microbial regrowth within the distribution system, the network of pipes delivering water to homes and buildings.
But even with a residual, things can grow in those pipes.
Oh, absolutely. Water distribution systems are massive, complex networks. They provide huge surface areas inside the pipes. And despite the residual disinfectant, microbes inevitably establish biofilms on those surfaces.
Biofilms like slimy layers.
Exactly. And microbes living inside a biofilm are much better protected from disinfectants than free-floating ones. Plus, the disinfectant residual naturally decreases the further the water travels from the treatment plant. So microbial growth in pipes is pretty much unavoidable.
What's the risk there?
The main concern is opportunistic pathogens. These are microbes that don't usually harm healthy people but can cause infections in those with weakened immune systems, the elderly, or other vulnerable individuals.
Like what kind of microbes?
Things like non-tuberculous mycobacteria, or NTM. Also Legionella pneumophila, which causes Legionnaire's disease. Pseudomonas aeruginosa is another common one, and even some free-living amoebae like Naegleria or Acanthamoeba. These can all thrive in pipe biofilms.
And I read something really interesting about those amoebae, those protozoa. They act like hiding places.
Yes, this is a really key point. These grazing protists munch on bacteria in the biofilms. But certain bacteria, including Legionella, Pseudomonas, and Mycobacterium species, have actually evolved ways to survive inside these protists.
Like Trojan horses.
Kind of. Being inside a protist shields them from disinfectants, helps them survive harsh conditions, and some research suggests it might even prime them, make them more virulent, or better able to infect human cells when they get the chance.
The showerhead example comes to mind, Legionella growing in there.
Exactly. Showerheads are notorious spots for biofilm growth, often enriched in NTM and Legionella. When you shower, the aerosolized water droplets can carry these bacteria right into your lungs. It's a perfect example of how our building plumbing becomes part of this microbial ecosystem, sometimes with health consequences, especially if the infrastructure is old or poorly maintained.
Which leads us directly inside our homes and buildings. We think of indoors as separate, maybe cleaner, but microbes are everywhere there too.
Everywhere. In the air we breathe, settled in dust, on every surface, inside the ventilation systems and the plumbing. We spend, 90% of our time indoors. We're constantly surrounded by and interacting with a really complex indoor microbiome.
And this has health implications, both good and bad, like that hygiene hypothesis idea.
Right. The idea that maybe being too clean, especially early in life, deprives our immune system of the microbial exposures it needs to develop properly, potentially contributing to rises in allergies and autoimmune diseases. It's a hot area of research.
How do we even know what microbes are indoors?
Mostly through genetic sequencing, looking for the 16S rRNA gene in bacteria and the 18S rRNA gene in fungi and protists. We can sample the air, collect dust, swap surfaces, and identify the communities present.
And what's surprising is that the indoor community isn't just a reflection of outside, is it?
Not entirely. While outdoor air definitely influences indoor air, especially fungi, which can reflect local geography, the bacterial communities indoors are often strongly shaped by the occupants.
Us and our pets.
Exactly. Human skin microbes, microbes associated with our gut or mouths, they become a major component of indoor dust and surface microbiomes. If you have pets, you can often tell just by looking at the microbes present, certain bacteria are strongly linked to dogs or cats. Rural homes might show signatures from farm animals.
So the indoor microbiome is like a fingerprint of who lives there.
In many ways, yes. Studies have shown that the microbes on surfaces like floors, doorknobs, kitchen counters closely resemble the microbes found on the hands, feet, and even noses of the people living there. It's so specific that the microbial signature of a house can often predict which family lives there, and it changes when people move out and new people move in.
That sounds like something out of CSI, forensics using microbes.
It's definitely heading that way. People are exploring combining microbial fingerprinting, the 16S data, with analyzing the chemical residues people leave behind, using metabolomics. Think about it. Maybe you could swab a phone and find not only the owner's skin microbes, but also traces of caffeine metabolites if they're a coffee drinker. It opens up some really interesting possibilities for forensic science down the line.
And our cleaning habits affect this indoor world.
Profoundly. The constant use of antimicrobial soaps and cleaners definitely shapes the indoor microbiome. The worry is that it might be selecting for more resistant microbes. For instance, many common disinfectants are less effective against certain gram-positive bacteria, so using them might inadvertently enrich for those types, potentially including antibiotic-resistant strains like MRSA. Our attempts at hygiene could have unintended consequences.
What about public spaces?
Yeah.
Like the subway.
Those are fascinating melting pots. You get a mix of outdoor air microbes, soil tracked in, but also a huge signature from all the people passing through: skin flakes, microbes shed from breath, hands touching surfaces. Subway air, for example, often has a strong signal of microbes typically found on human feet and hands.
Yikes. And speaking of plumbing again, toilets.
Yeah, flushing a toilet creates a plume of aerosolized particles, potentially containing microbes from the waste. It's another way enteric pathogens could potentially be dispersed within a building, especially in public restrooms. Now, besides just living with us indoors, microbes can also actively attack the materials we build with. Microbially influenced corrosion, or MIC.
Right. This isn't just normal rust or decay. It's microbes making it happen faster.
Exactly. They can accelerate the corrosion of metals, stone, concrete, causing billions of dollars in damage every year to pipelines, bridges, buildings, infrastructure.
Metals seem particularly vulnerable, especially iron pipes and things.
Very much so. MIC is a major headache for iron and steel infrastructure, particularly in environments without much oxygen.
And the main culprits are those sulfate-reducing bacteria again, SRBs.
SRBs are definitely prime suspects in many cases of anaerobic metal corrosion. But other groups can be involved too: iron reducers, iron oxidizers, even methanogens.
How do the SRBs corrode the metal? We talked about the H2S they make being corrosive.
Right, the hydrogen sulfide product is chemically corrosive itself. That's the souring issue in oil pipelines if sulfate gets introduced, like through seawater injection used to maintain pressure. One way to sometimes control SRBs in oil fields is actually to inject nitrate. Other bacteria use nitrate and can outcompete the SRBs for food, plus some nitrate reducers can oxidize the corrosive sulfide.
But can SRBs attack the metal more directly?
It seems so. Two main mechanisms are thought to be involved beyond just the H2S. One is indirect. When iron corrodes chemically, it produces hydrogen gas, H2. SRBs gobble up this H2 as an energy source. By constantly removing the H2, they effectively pull the corrosion reaction forward, making it happen faster.
Okay, that makes sense.
The other, more recently understood mechanism is direct electron uptake. Some SRBs, like disulfopylocorrodins and even some methanogens, seem to have ways to physically attach to the metal surface and pull electrons directly from the metallic iron, friend zero, using it as their energy source. This directly oxidizes the iron, causing it to corrode.
So they're literally eating electricity from the metal.
In a manner of speaking, yes.
And it's not just metals. Stone and concrete can be damaged too. Biodeterioration.
Absolutely. Microbes colonize stone surfaces, even living inside the pores endolithic communities. You get bacteria, archaea, fungi, algae, cyanobacteria. The photosynthetic ones, like algae and cyanobacteria, often form the base of the food web, providing nutrients for others.
How do they break down something as hard as stone or concrete?
Fungi are often key players here. They can produce organic acids, like oxalic acid, which slowly dissolve the minerals in the stone or concrete matrix. This creates microscopic pits and increases porosity, making the material weaker and more susceptible to physical weathering like freeze-thaw cycles. They also produce pigments for UV dew protection, which can discolor surfaces.
And there's that really dramatic example in sewer pipes crown corrosion.
Yes, that's a severe form of concrete biodeterioration. It happens in the headspace of concrete sewer pipes above the wastewater flow and can actually lead to pipes collapsing.
How does that work? Is it SRBs again?
SRBs start the process, but indirectly. Down in the anaerobic wastewater, SRBs produce hydrogen sulfide, H2S gas, from sulfate and organic matter. This H2S gas escapes into the air-filled headspace above the water.
Okay.
In that humid, oxygen-rich headspace, different bacteria take over. Sulfur-oxidizing bacteria. First, types that like neutral pH start oxidizing the H2S. But as they produce acid, conditions become favorable for acid-loving sulfur oxidizers. These guys convert the H2S all the way to sulfuric acid, H2SO4.
Sulfuric acid dripping onto the concrete roof of the pipe.
Exactly. And a sulfuric acid is extremely corrosive to concrete. It reacts with the calcium hydroxide, lime, and other cement components, forming minerals like gypsum. As these minerals form and expand within the pores of the concrete, they create internal pressure, causing cracking, spalling, and eventually total structural failure. It's a huge problem for aging sewer infrastructure.
Wow. So reflecting on all this, we've gone from microbes mining metals for us, to cleaning up our messes, to shaping our indoor air, living in our water pipes, and even eating away at the structures themselves. It's quite a journey.
It really is. This deep dive shows that our built environment isn't just inert bricks and mortar and pipes. It's a dynamic microbial habitat. And understanding these interactions, I think, gives us a whole new lens on everything from pollution and corrosion to public health and even forensics. We see the problems, but also the potential solutions microbes offer.
It definitely hammers home how interconnected we are with this invisible world, even in places we think of as completely human-made and controlled.
Absolutely. Maybe the final thought to leave people with is this. Even in what seems like the most sterile, controlled indoor space, we are constantly sharing that space with a vast, diverse microbial community. A community that's partly shaped by us, reflects who we are, and in turn, subtly or dramatically influences our health and surroundings in ways we're really just beginning to fully appreciate. What part of this hidden landscape stands out most to you?